Namaste and welcome to the channel. Today we are going to explore one of the Ayurveda's classical herbo-mineral formulation, that is Hinguleshwar Rasa. This medicine has been traditionally described in Ayurveda text for the management of the conditions such as Jwara, Vati and certain infectious disorders. In this video, we don't rely on myths or any opinions. Instead, we will examine what scientific research says about Hinguleshwar Rasa. We will discuss published studies on its antibacterial activities and experimental research in Vati and its Ayurvedic pharmacodynamic in the management of fever. If you are an Ayurveda student, researcher, healthcare professional, or simply interested in evidence-based Ayurveda, this video is for you. So without further delay, let's begin our journey into the science and traditional wisdom behind the Hinguleshwar Rasa. Here, Hinguleshwar Rasa is reference from the Bhaishajya Ratnavali, Jwara Chikitsa Prakarana. The verse 361, Tulyansham mardayat khalle Pippalim Hingulam visham Gunjardham madhunam deyam Vata Jwara nivruttaye. Here, use ingredients Pippali, Hingul and visham means Vatsanabha, taken equal quantities, and triturate all these three ingredients together in a Khalva Yantra using water to form a fine homogeneous mass, and prepare Vati weighing about half Gunja, approximately 62.5 mg. Dosage half Gunja, Anupana, Madhu, and indication is Nutan Vataja. Now we will see the research article about Hinguleshwar Rasa. First, evaluation of Hinguleshwar Rasa by ICP AES means elemental qualitative analysis for standardization. This is the Ayurvedic herbo-mineral formulation, and Rasa contains purified minerals and herbal ingredients including Hingul, Vatsanabha and Pippali. The author states standardization of this formulation at the elemental level is also necessary in the modern era to ensure its quality, consistency and safety. ICP-AES was selected because it can direct trace and heavy metals accurately, and the study also investigates elemental changes occurring after purification and formulation preparation. This study was conducted because Ayurvedic purification is believed to alter the properties of minerals and herbs, and modern analytical techniques are required to validate these traditional Ayurvedic procedures, and standardization improves quality control and supports wider acceptance in Ayurvedic medicines. So researcher intended to evaluate elemental composition before and after purification, detect elemental changes during the preparation of Hinguleshwar Rasa, and scientifically validate traditional purification procedure. Use ICP AES as a tool for standardizing the formulation. Here the objective to study where to purify Hingul and Vatsanabha according to classical Ayurvedic methods, and to prepare Hinguleshwar Rasa using purified ingredients, to perform ICP-AES elemental qualitative analysis of raw Hingul, purified Hingul, raw Vatsanabha, purified Vatsanabha, Pippali and Hinguleshwar Rasa, to compare elemental composition before and after purification, to determine whether purification and formulation preparation produce elemental changes, to contribute towards standardization of the formulation. Now we see the detailed methodology and experimental laboratory study involving Ayurvedic pharmaceutical preparation and elemental analysis. First step is a raw herbs and mineral material were identified using physical characteristics and botanical parameters. Step second is a purification of Hingul. Hingul was triturated with lemon juice 7 times. Material was washed 7 times with deionized water. Total purification period about 14 days. The purified Hingul becomes smooth, dark red, lusterless and nearly neutral in pH. Step third is the purification of Vatsanabha. Small pieces of Vatsanabha were immersed in fresh Gomutra. Gomutra was replaced daily. Treatment continued for 3 days in sunlight. Outer covering was removed and purified material was dried in sunlight. Step fourth is a preparation of Hinguleshwar Rasa. Equal quantities purified Hingul, purified Vatsanabha and Pippali powder were mixed and triturated with a stone grinder until a homogeneous mixture was formed. Step 5 for the ICP-AES analysis. Sample analyze in 6 sample total. that is a raw hingul, purified hingul, raw Vatsanabha, purified Vatsanabha, Pippali and Hinguleshwar Rasa. And sample prepared for ICP AES, 0.1 gram sample use, digest with concentrated nitric acid, microwave digestion at 190 degrees Celsius, diluted to 50 ml and introduced into the ICP AES instrument for elemental analysis. Now we see the detailed results. First result, raw versus purified hingul. Raw hingul contains 19 detected elements and purified hingul contains 21 elements newly detected after purification, that is a chromium, sodium and tungsten. These elements were absent in raw hingul. Result second is a raw or versus Vatsanabha. Both raw and purified Vatsanabha show essentially the same elemental profile. No significant elemental differences were observed after purification. Result third is a Hinguleshwar Rasa. The final formulation contained 19 detected elements. Some elements found in purified hingul or purified Vatsanabha such as chromium, gallium, molybdenum, nickel, lead, tungsten were not detected in the finished formulation. Here the interpretation of this result to suggest that purification of hingul alters the detectable elemental profile. Purification of Vatsanabha did not produce major detectable elemental changes. Mixing and trituration during the formulation preparation may further alter elemental composition, as some elements detected in purified ingredients were absent in the finished product. At the end of the concluded that ICP AES is a usually analytical technique for evaluating Ayurvedic formulations. Elemental changes may occur during purification or trituration. The study supports the use of ICP AES for standardization of Hinguleshwar Rasa and its ingredients. No unique or significant specific elemental mark was identified in the final Hinguleshwar Rasa formulation. Now we see the second article related Hinguleshwar Rasa, that is a evaluation of antibacterial activity of Hinguleshwar Rasa. In introduction, infectious disease remains a major cause of illness worldwide. Antibiotics have greatly reduced mortality, but their excessive and inappropriate use has led to antibiotic resistance. The author explains that many bacteria no longer respond effectively to commonly used antibiotics. There is an urgent need to identify alternative antimicrobial agents. Ayurveda contains numerous classical formulations traditionally used for fever and infectious. Hinguleshwar Rasa has long been used in Ayurvedic practice, but modern scientific evidence supporting its antibacterial effect is limited. Therefore, laboratory evaluation was necessary to determine whether this traditional formulation possesses measurable antibacterial activity. Why this study was conducted? Because few scientific studies have validated the antibacterial properties of Hinguleshwar Rasa. Classical Ayurvedic texts describe its usefulness in infectious condition, and experimental evidence is needed to support its traditional use. The research aimed to bridge traditional Ayurvedic knowledge with modern microbiological testing. The main objective was here to evaluate the in vitro antibacterial activity of Hinguleshwar Rasa prepared according to the Ayurvedic Formulary of India or Bhaishajya Ratnavali against common pathogenic bacteria using the agar-well diffusion method. In detail methodology, study design is here as an experimental and in vitro laboratory study. No human or animal subject were included in it. First step is a preparation of the drug. Hinguleshwar Rasa was prepared according to classical Ayurvedic procedures described in Bhaishajya Ratnavali and AFI. Formulation was prepared using standardized purification and pharmaceutical methods before microbiological testing. The test organisms are used here. 5 important bacterial pathogens tested here. First is a Streptococcus pyogenes, second is a Staphylococcus aureus, third Escherichia coli and Salmonella typhi, and fifth is a Pseudomonas aeruginosa. These organisms are commonly associated with fever and infectious disease. Antibacterial testing method use agar-well diffusion method. Procedure is mentioned here. Nutrient agar plates were prepared. Each bacteria stain was cultured. Bacterial suspension was spread uniformly on the agar. Wells were made in the agar using a sterile borer. Different concentration of Hinguleshwar Rasa solutions were added into the wells. Streptomycin serves as the positive control also. Plates were incubated and the zone of inhibition around each well was measured. A larger inhibition zone indicates stronger antibacterial activity. Now we see the detailed result. Found that the Hinguleshwar Rasa showed antibacterial activity against all tested bacterial strains. Activity differed depending on the bacterial species and the concentration tested. Highly concentration generally produce larger inhibition zones. Streptomycin produce greater inhibition than Hinguleshwar Rasa as expected for a standard antibiotic. Nevertheless, the formulation demonstrates measurable antibacterial effects supporting its traditional use. The authors primarily reported comparative inhibition pattern rather than claiming superior to standard antibiotics. The final conclusion is that Hinguleshwar Rasa possesses in vitro antibacterial activity against several clinical important pathogens. The findings support traditional Ayurvedic claims regarding its use in infectious disease and fever. The medicine may have potential as an complementary antimicrobial agent, but laboratory findings alone are insufficient to establish clinical sufficiency. So additionally, scientific investigations are required before therapeutic recommendation can be made. Future scope for this study to isolate and identification of the active antimicrobial components, determination of the minimum inhibitory concentration and minimum bactericidal concentration, also study to clarify the mechanism of antibacterial action, also toxicity and safety evaluation, animal experiments and well-designed clinical trials in patient, and standardization and quality control studies to ensure consistent preparation. Now we see the third article. Title is the in vivo histopathological efficacy study of Hinguleshwar Rasa and endomycin on FCA induced rheumatoid arthritis in palm model of rats. In introduction, rheumatoid arthritis is a chronic autoimmune inflammatory disease characterized by persistent synovial inflammation, joint swelling, pain, cartilage destruction and bone erosion. If left untreated, it leads to deformities and disabilities. According to Ayurveda, the clinical features of rheumatoid arthritis resembles amvata, a disease caused by the accumulation of ama together with aggravated vat dosh. Classical Ayurvedic text, particularly Rasa Tarangini, recommended Hinguleshwar Rasa for the management of amvat. Although Hinguleshwar Rasa has been used traditionally for many years, there was little scientific evidence demonstrating its anti-inflammatory action or its ability to restore damaged tissue or joint tissue. Therefore, the researcher designed an experimental study using an established rat model of rheumatoid arthritis to scientifically validate its traditional therapeutic claims. The study was undertaken for several reasons. Because rheumatoid arthritis remains a chronic disabling disease requiring long-term therapy. Conventional NSAIDs such as endomycin reduce pains and inflammation but may produce adverse effect during prolonged use. Hinguleshwar Rasa is widely prescribed in Ayurveda for amvata. Its scientific validation of its efficacy was limited. Histopathological evidence demonstrating actual tissue healing was lacking, and the researcher aimed to compare the traditional medicine directly with a standard modern anti-inflammatory drug. Thus, the study sought to provide experimental evidence supporting the Ayurveda use of Hinguleshwar Rasa in inflammatory joint disease. The primary purpose of this study was to evaluate the anti-inflammatory and histopathological efficacy of Hinguleshwar Rasa in rats with FCA-induced rheumatoid arthritis and compare its therapeutic effects with those of endomycin. The objective includes to induce rheumatoid arthritis in rats using FCA, to administer Hinguleshwar Rasa after arthritis had developed, to compare its therapeutic efficacy with endomycin, to assess reduction in paw swellings as an indicator of anti-inflammatory activity, to examine microscopic changes in ankle joint after treatment, to evaluate restoration of normal joint architecture following therapy, to determine whether Hinguleshwar Rasa possesses disease modifying potential in experimental rheumatoid arthritis. Now we see detailed methodology is here. Study design, experimental or comparative in vivo animal study. Laboratory rats are used here. And disease induction. Rheumatoid arthritis was induced by injecting FCA, means prion complete adjuvant, into a hind paw, and arthritis developed over approximately 14 days. Experimental group rat are divided into group included normal control, arthritic control, and standard treatment group, that is induced endomethacin, and test treatment group, that is induced Hinguleshwar Rasa. Treatment schedule. Arthritis was induced on first day. By day 14, arthritis had developed. From day 15 to day 28, animals received Hinguleshwar Rasa and endomethacin by the oral route. Assessment. The anti-inflammatory effect was evaluated by measuring paw swelling throughout the experiment and comparing reduction in edema among the group. And histopathological examination. At the end of the 28-day study, animals were sacrificed, ankle joints were collected, tissue samples were fixed, processed, and sectioned and stained. Microscopic examination assessed synovial inflammation, congestion of blood vessels, tissue degeneration, cartilage integrity, bone architecture, and restoration towards normal histology. Histopathological findings were compared across the normal control, arthritic control, and Hinguleshwar Rasa and endomethacin group. The result is here. Reduction in paw swelling. Both treatment group demonstrated significant reduction in paw edema compared with untreated arthritic rats, indicating anti-inflammatory activity. And in histopathological findings, arthritic group control. A microscopic examination showed marked inflammatory changes, joint tissue damage, and vascular congestion and pathological alteration in bone and surrounding tissue. In Hinguleshwar Rasa group, microscopy demonstrated the substantial recovery towards normal joint histological architecture and reduced inflammatory infiltration, minimal pathological changes, decreased vascular congestion, and restoration of tissue organization. Third is a endomethacin group. The endomethacin treated animal also exhibits near normal histological appearance, reduction of inflammation, and improved joint morphology. Comparative findings. Both the treatment were effective. However, the investigators reported that Hinguleshwar Rasa was showed better restoration of joint histopathological changes caused by FCA induced arthritis than endomethacin in the experimental model. At the end of the conclude that Hinguleshwar Rasa possesses significant anti-inflammatory activity in the FCA-induced rat model of rheumatoid arthritis. It promotes restoration of normal joint histology and reduces pathological changes associated with arthritis. Its effects were comparable to and in the reported histopathological assessment appear better than those of endomethacin. These findings provide experimental support for the traditional Ayurvedic use of Hinguleshwar Rasa in Amwat. It is important to note that these results come from an animal study and do not by themselves establish superior effectiveness in humans. The clinical importance of the article. This study was several important implications. It provides experimental evidence supporting the traditional Ayurvedic indications of Hinguleshwar Rasa for Amwat. The observed reduction in inflammation and improvement in joint tissue architecture suggest potential anti-inflammatory and tissue protecting properties. Histopathological recovery indicates possible benefits beyond symptomatic pain relief, although confirmation in humans is needed. The comparison with endomethacin offers a useful benchmark against a standard NSAID in a preclinical setting. The findings support further pharmacological, toxicological, and well-designed clinical studies to determine safety, optimal dosing, and efficacy in patients with rheumatoid arthritis before routine clinical use can be recommended. Now, last we see fourth article. Title is the effect of Hinguleshwar Rasa in Jwara. That is mode of action and pharmacodynamics in Jwara is explained in this article. Author described Jwara is one of the most important diseases in Ayurveda because it's affect the body, sense and mind. According to Ayurvedic pathology, fever commonly begins with mandagni. Weak digestion results in the formation of ama. Obstructs body channels, disturbs the doshas and ultimately produces fever. In Vatajwar and Amjwar, treatment should promote Dipan, Pachan and Dosha Shaman. The paper notes that Hinguleshwar Rasa is a classical herbo-mineral formulation, indicates for such a condition and seeks to explain its therapeutic rationale using classical Ayurvedic pharmacodynamics. The review was undertaken because Hinguleshwar Rasa is widely used for fever in Ayurvedic practice. The formulation contains only a few ingredients but is traditionally considered highly effective. There was a need to explain how the medicine works based on the pharmacodynamic properties of each ingredient. Author aimed to connect traditional Ayurvedic concepts with available modern pharmacological knowledge. Thus, the study provides theoretical scientific basis for the classical indication of Hinguleshwar Rasa in Jwara. The principal purpose was to explain the probable mode of action of Hinguleshwar Rasa in fever, to analyze the pharmacodynamic properties, that is Rasagun, Virya, Vipak and Karma of its ingredients, to describe how these properties interrupt the pathogenesis of Amjwar and Vatajwar. The objectives here: to review the classical composition of Hinguleshwar Rasa, describe Rasa Pachak of each ingredient, explain individual pharmacological role of Shuddha Hingul, Shuddha Vatsanabha and Pippali, explain the combined pharmacodynamic profile of the formulation, illustrate how the formulation breaks the pathogenesis of fever, correlate Ayurvedic concepts with the available modern pharmacological evidence. Now we see the methodology. Because of this is a review article, no laboratory experiment or clinical trial were performed. Instead, the authors carried out a literature-based analysis by reviewing classical Ayurvedic texts including Rasa Tarangini, Rasa Ratna Samuchay and Dravya Gun Vignan, examining the composition and purification of the ingredients, analyze each ingredient according to Rasa Panchak, Rasa Guna, Virya, Vipak and Karma, integrating classical description where reported modern pharmacological activities and antimicrobial to explain the formulation likes mechanism in fever. The result conclude that the combined pharmacodynamic profile of Hinguleshwar Rasa supports its traditional use in fever. First is a Shuddha Hingul. After proper purification, Hingul is described as a Jwaragna, Tridoshhar and Vishahar. It is considered to enhance the therapeutic action of the formulation and contribute to fever relief. Shuddha Vatsanabha, following purification, Vatsanabha is described as a possessing Ushna, Virya, Dipan, Pachan, Vat, pacifying activity. The review also notes reported modern pharmacological properties such as analgesic and anti-inflammatory effect after appropriate processing, which may help reduce fever associated pain and inflammation. In Pippali contributes Dipan, Pachan, Rasayan and immunomodulatory and anti-inflammatory actions. Its active constituents piperine is discussed as enhancing bioavailability and supporting the absorption and action of the formulation. Combined pharmacodynamics, the formulation as a whole predominantly exhibits Katuras, Laghuruksha and Tikshnagun, Ushnavirya and Katuvipak. These properties are considered to stimulate impaired Agni, digest ama, clear obstructed channel, pacify Vata and Kapha and reduce fever and restore physiological balance to breaking the disease processing in Amjwar and Vatajwar. The final conclusion that Hinguleshwar Rasa possesses logical Ayurvedic pharmacodynamic basis for its use in fever. The combined properties of purified Hingul, purified Vatsanabha and Pippali collectively supports Dipan, Pachan, Jwaragna, Tridosh balance, especially Vata and Kapha, interrupted of the pathogenesis of the Amajwar and Vatajwar. The review emphasizes that these conclusions are based on classical literature and pharmacodynamic interruption rather than direct experimental or clinical evidence. Clinical importance of this study to provide scientific and Ayurvedic rationale for the traditional use of this kalpa in fever. explain formulations mechanism through Rasa Panchak and Samprapti Vighatan to understand for students and practitioners of Ayurveda, highlights the complementary roles of purified Hingul, purified Vatsanabha and Pippali in managing fever associated with impaired digestion and ama, suggests possible links between classical Ayurvedic concept and modern pharmacological actions such as anti-inflammatory, analgesic, anti-modulatory and viability enhancing effect. Identifies the need for future experimental studies and well-designed clinical trials to validate these proposal mechanism and confirm clinical efficacy and safety of the patient. As we have seen throughout this video, Hinguleshwar Rasa is a classical Ayurvedic formulation with a long history of traditional use. Recent laboratory studies, animal research and pharmacodynamic reviews suggest promising antibacterial and anti-inflammatory properties and provide a scientific basis for some of its traditional indication. Because Hinguleshwar Rasa is a herbal mineral medicine, it should only be used under the guidance of a qualified Ayurvedic physician with properly prepared and quality control formulation. If you found this video informative, please like, share and subscribe to the channel for more evidence-based discussion on Ayurveda, medicinal formulation and current scientific research. Thank you for watching. Stay healthy, stay informed, and I will see you in the next video. Namaste.